mechanism for its RNA, right? It has its a repair mechanism for its RNA. So its mutation rate is not as fast as you would see with HIV, for instance, or with the other RNA viruses. So initially, when studies on vaccines and vaccine development started, we thought this was actually quite a big benefit because you would have a vaccine that that would last for quite a bigger time. And time has proven us wrong. It's quite a very clever bug, this one. It also has the envelope, an envelope around it. If you remember from your basic microbiology at medical school, the viruses are either naked or they've got a coat. The coat is the envelope. It's a lipid envelope. And what is the importance of this envelope? The importance of this envelope actually it tells us that this virus is vulnerable to desiccation and disinfectants. So it's a virus that cannot persist for a very long time in the environment. And then two, it's highly susceptible to your alcohol disinfectants and your normal disinfectants. And then it's got a membrane cover and the now notorious spike protein. Now the spike protein is the protein that it uses to attach to the receptors on the host. And the commonly used receptor is the ACE2 receptor, that's the angiotensin converting enzyme 2 receptor. And this enzyme is promiscuously available throughout the body. It's in your GIT tract, it's in your kidneys, it's in your lungs, it's in your nasopharynx. And that's the disaster with this virus. It's got a large sample space of receptor field to choose from. From an immunological point of view, what do we know, right? Earlier on in the outbreak, they took a lot of epitopes that had been collected based on the earlier SARS outbreaks, that is the MERS outbreak and the SARS in 2022. And so when you took these pools and you tested them for CD4 and CD8 responses amongst patients that had recovered from COVID, it was noted that 100% of these patients had epitopes, had a CD4 response. So they had IgG antibodies, 100% of them against the spike protein, and then IgA, which is quite important for your mucosal immunity, against spike protein 2. And this is what gave the direction for people in vaccine development to start targeting the spike protein. Not only that, but there were also T-cell responses, right? And the T-cell responses were actually much more widespread. They were against the nucleocapsid, they were against the membrane protein, and they were against the spike protein as well. So there is a CD8 factor that comes into the immune response to SARS-CoV. And then further testing in non-exposed individuals actually detected these T-cell responses as well. Now, this can be associated with the coronaviruses that we get, our GIT infections, those ones that will cause you a diarrhea, a mild diarrhea, and those coronaviruses that are associated with the common cold. So the clinical impact or the clinical benefit from this cross reactivity is still unknown at this stage, and it's still work in progress. But then still we've got challenges from this knowledge of immunology, because we still don't know much about the correlates to protection. What are the correlates that we can use to say, for instance, the vaccine protects you against infection, acquiring the primary infection, or the IgA response that allows you to deal with the virus at your mucosal level, and then you cannot spread it, you cannot transmit it to others. And what are the correlates that we can use to determine disease progression? Or what can we use to predict what that subset of patients that subsequently end up hospitalized or they die? Now, vaccinology has moved in the last last 40 years, and particularly since the 1980s, when we had the hepatitis B vaccine, which was the first recombinant vaccine. So since 1986 thereabouts and to date, you can see the technology has moved, right? It's becoming more towards vaccines that are based on recombinant proteins, that are based on molecular modification, that is DNA or your RNA. And you've also had development of adjuvants in the process. What is an adjuvant? An adjuvant is an accessory to the antigen that allows the immune response to shoot several times higher. And what do we see? How have we used this for COVID-19 vaccines? You can now see that we've got about 63 vaccines now that are in clinical development, and even more that are still in the preclinical stage. You've got about 179 vaccines that are in the preclinical stage. And most of these vaccines are using the new technologies, right? So these protein subunits, the recombinant subunits, that's 32% of the vaccines coming in. And the viral vectors, the non-replicating viral vectors, the adenovirus, for instance, it's about 16%, and the DNA in the RNA viruses as well. Now, just a quick note on this new technology. The major vaccines that have come in have utilized the mRNA approach and the non-replicating virus vector approach. What happens with mRNA? Over time, people have learned to preserve mRNA. It degrades easily, it gets contaminated easily. So technology has allowed it to you get mRNA from a baculovirus or another virus, for another virus platform, harvest this mRNA and put it in a transport mechanism, which is usually a lipid nanoprotein. And you inject this into the host, right? What the mRNA gets delivered to the host ribosomes. It's all outside the nucleus in the cell cytoplasm. And then it is transcribed. It is translated into proteins. And these proteins are the spike protein that elicits an immune response. Then you develop neutralizing antibodies. It's quite simple. The other bit is the adenovirus, where you get an adeno, a harmless, not harmless really, you know, the common circulating adenoviruses, usually the non-pathogenic adenovirus serotype 26 or 20 or 5. And in it, you inject the viral DNA, right? The part of the DNA that codes for the spike protein gets incorporated into the adenovirus. And then the adenovirus is then used as a transport media. It becomes the vaccine. You inject it into the host. The host takes this non-replicating virus, and the virus does its infection process, gets into the nucleus, into the host DNA, and you get transcription and translating into mRNA into the viral protein. One of the side effects which become very important with the viral vectored infection, with the viral vectored vaccines, is already the host might have an immune response to the adenovirus, right? So there's a potential that you will naturally neutralize this vaccine. So that's why you need to have a booster doses for this. What is currently available? Right. What do we have? I'm touching on the vaccines that we have here in looking at basically here in South Africa. This one out of the post. Hello, is somebody?
Hello. Okay. Let me try to mute this person.
Okay, can you people go on mute? So what is the first one out of the gate was the Pfizer vaccine. The Pfizer vaccine is using mRNA technology, and in the clinical efficacy trials, if you looked at clinical infection, the prevention of mild to moderate disease, it was 95% efficacious. and its efficacy against hospitalizations, protecting those that got infected, got disease, but did not progress to severe disease needing hospitalization, was about 90%. How do you administer this vaccine? It's 2 doses, 21 days apart. What's the big drawback with this vaccine is it requires -70 storage, right? You store it at -70, you transport it at -70, but once you take it out for vaccination, it is stable at 2 to 8 degrees in ordinary fridge temperature for about 120 hours. The second one that came out in mRNA based was the Moderna, with same efficacy rates and same protections against hospitalization. In the interest of time, I'll actually then touch on 2, I think 3 that are quite interesting on this slide. There is then the AstraZeneca, which has caused a lot of debate. It's a viral vector vaccine. That is, it's based on the adenovirus. Right. Now, in clinical trials, there was varied protection, right? If you looked at it, depending on geographic region and depending on the dose. When they started the trial, they were using a standard dose for first inoculation and the second inoculation. And the first inoculation was noted to have lower efficacy at 67%. Than the lower dose, which gave efficacy at 90%. The reason being what I explained earlier on the natural neutralization of the vaccine by pre-existing antibodies, right? If you give it at higher dose. Right, so this AstraZeneca vaccine comes in two doses, and it was going to have a very big global impact because it is stable at 2 to 8 degrees. You can store it at 2 to 8 degrees, you can transport it to 8 degrees and vaccinate at that. The other vaccine, a viral vectored vaccine, is the Johnson & Johnson vaccine. And this has been very interesting because if you look at a protection from infection in the clinical trials from the South African centers, it only had protection of about 57% if you looked at acquisition of infection. But it was efficacious in protection against hospitalization. That is, in protection against severe disease with 85%. And this was across all the clinical trial centers, both in South Africa, in Europe, Brazil, and the United States. And the beauty of the Johnson & Johnson, it's a single dose, and it also stores at the same temperatures as the AstraZeneca. Sputnik is interesting for other scientific reasons because you are looking at heterologous adenovirus injections here. And Sinopharm is an inactivated virus vaccine, and it's from China. From the clinical trials in China yet to be published, you see efficacy rates ranging between 50 to 70%, and it's also a 2-dose vaccine. Right. So if you are going to then choose a vaccine, what are the considerations that you want to pay attention to? It's the safety, right? It's a vaccination, you are vaccinating healthy people. So safety is very paramount. It's the efficacy, and particularly in the populations the high-risk populations that we are talking about, the cost, the transport and storage, and the dose regimen. And what we have learned recently with COVID is you need very good surveillance programs. Right, surveillance programs to pick up changes in your strains that are circulating, emergence of variants that can affect vaccine efficacy. Because vaccines have a core business, and their core business is to survive, and to do that they mutate, and they also recombine, and that's how we get new viruses and expanded host ranges for viruses. And what have we learned with the COVID variants? Right, first, right early in the beginning, we had the D614 mutation that happened in China in March 20. Right, what the impact of this change in the virus was, it increased transmissibility. It became more efficient in binding, right, and then became more transmissible. And it is this mutation that caused it to become a global phenomenon, to be so widespread. Then within a short time, in September 2020, there was the 4531, right? And then that mutation was in Denmark and the Netherlands, right? We saw a change here where the virus actually became adept at infecting the ACE2 receptors in mink, right? So you now had human to mink transmission, mink to mink, and mink to human. And the implication of this from a public health point of view is you are creating an animal reservoir. So whatever you do to this virus, as long as there is a reservoir in the animal, it will jump back into humans. So it has resulted in massive culls on mink farms in Denmark and in the Netherlands. Then we had lineage B117 from the UK. Now, this mutation also, this lineage has a number of mutations. That's why we call it a lineage. About 23 of them, and 17 of them are actually the important ones. And they have resulted in spreading 56% times faster than the normal variants. And it has higher affinity for the receptors. And what we think is it's a reflection of the magnitude of community spread and a chronic infection, giving the virus an opportunity to select for survival. And then we've got the South African variant, B351. Now, it detected during this current second wave, and it was quite fortuitous because it was at the same time that the clinical trials for the AstraZeneca vaccine, the Novavax vaccine, and the JJ vaccines were happening in South Africa. And it has become the 80% of the circulating strains in South Africa with high viral loads in the specimens. And what it means is you've got higher transmission rates. And it has got two worrying mutations, right? And these mutations have been picked up in the UK lineage as well. That is 417N and 484. And these two mutations allow the vaccine to escape immunity, right? So it will escape natural immunity. That means we're now seeing patients that were infected in the first wave coming in sick in the second wave. And there is now evidence of this variant affecting viral efficacy. And like I was saying, the impacts of variants developing, they tend to spread faster, they become more transmissible. We don't have evidence of increased mortality or more severe pathogenesis in the new variant patients. But what's quite interesting, why I put up this slide, is the politics that's associated with these bugs. The British started saying it was the South Africans who came up with this, and then the South Africans had to issue a media statement to say, no, we are not responsible for this. It's actually in the behavior of the virus. And what's quite nice is we noticed this variant when the trials were actually happening. I put this slide because of what the Africa CDC has said, that about the AstraZeneca vaccine, that you can proceed to use it if you don't have a variant that's found in South Africa. But how many countries have got surveillance mechanisms to know what are the circulating variants in their communities? I'll have to go a bit fast in the interest of time, but are viral vector vaccines effective against the variant that we have seen here? In South Africa, we know AstraZeneca vaccine efficacy goes down up to 22% when you are confronted with this variant. The Johnson & Johnson vaccine seems to do better. The efficacy goes down to 57% and 72% in the US, but we'll see the US will revise downwards as they get these variants in their population now. Now, the Johnson & Johnson vaccine seems to protect against hospitalization in 80% of these patients that are infected with the new variant. In the laboratory, if you do neutralization assays, you will see that the antibodies go down in the AstraZeneca vaccine by about 86-fold. So it's actually quite severely affected by this new variant. Now, the protein subunit vaccines, one of the vaccines that is in the pipeline is the Novavax vaccine, and that was on clinical trial here recently. And during the trials, when we were having this new variant, overall efficacy down, bomb, 49%. Right. Now, if we look across all the vaccines and from laboratory studies, we don't have much studies on the Johnson & Johnson in the laboratory, but we know the Pfizer and the Moderna, the Pfizer vaccine, the mRNA vaccine, neutralization antibodies go down threefold. There is no data yet when it comes to the clinical efficacy. We know Moderna goes down 6, AstraZeneca up to 86. So it actually falls out when when you do laboratory studies. Clinically, what does that mean, right? Johnson & Johnson is still okay because in terms of efficacy against severe disease and against hospitalization, we still have something in the arsenal. We don't have clinical data for Moderna and Pfizer. AstraZeneca, clinically in mild and moderate disease, 20%, 22% efficacy. But what we don't know is the level of protection in severe disease and in disease that requires hospitalization. That's why the rollout program was stopped, and it's now been introduced again in a stepped up kind of approach to see whether it has an impact in severe or hospitalized disease. We still need to move further with Novavax vaccine, its efficacy sitting at 49%. We now need to elucidate what's that impact going to be in severe and in hospitalizations. So what we have in summary and what we know about the vaccine against what is happening in the strain here in South Africa is there is laboratory evidence that there is a reduction in vaccine-induced antibodies. And that reduction varies from minimal with the mRNA vaccines to quite substantial, particularly if you look at the AstraZeneca vaccine. Right. Now, we've got clinical trial evidence now of reduced efficacy in mild to moderate disease for AstraZeneca vaccine, 22%, Novavax at 49%, and the JJ at 57%. But we still need data for the mRNA, and we still need data. Right. And we still need data against hospitalization. Now I'm on my final slide, and as a clinical micro, I won't do this justice without talking about diagnostic testing, right? What is the impact of genetic variants on diagnostic testing? Currently, it seems to be minimal, but if your laboratory systems are using the targets based on the S gene, be very careful. You need now regular monitoring to see the performance of your laboratory tests against these variants, because you will end up with a lot of false negatives, right? You will have people with clinical disease with the laboratory saying, no, there is no evidence of COVID. So in conclusion, really, we will see these variants continue to emerge as we get widespread community infection. The impact of these variants on antigenicity, vaccines, and the clinic is still unpredictable. So you will need very active molecular surveillance so that you know what strains are circulating in your population. And the vaccine manufacturing approaches need to be agile enough to be able to change the genetic formulations of the vaccines to cover the new or the emerging variants in the same way that you do with the flu vaccine. And the biggest lesson that has come from the South African vaccine program is you need actually a phased approach to vaccine introduction, right? Particularly where you've got variants that are becoming predominant, right? You want to know efficacy at different time points, at infection, for severe disease, for hospitalization, and ultimately, is it effective in preventing death? Thank you, and my apologies for taking a bit longer than I should have.
Thank you, Becky. Thank you very much for the very, very impressive presentation, very scientific. I think we would straight away go into questions so that at least we try to preserve some time. I think from the charts, Dr. Chirisa, Macron Chirisa, you have a question. Please, can you go ahead and ask the question?
Thank you very much, colleagues, for this very wonderful webinar, and thanks, Becky, for such an insightful presentation. My question was just on I was focusing, on when you're looking at the vaccines in terms of what we are looking at introducing in our own country in the next few weeks. That's the Sinopharm and the Sputnik vaccine, but specifically the Sinopharm, which is an attenuated virus. And I was just wondering what impact
Dr.
that would have
Male, we
in
can't
terms
hear
of.
Mark John.
Oh, okay. Let me start again. What about now? Can you hear me, Dr. Maulana?
Hello, is everyone hearing
Yes,
what Mark?
Yes,
Okay.
yes, we
We
can
can
hear
hear you. We can hear you.
Okay, fine.
Yes,
Okay.
we can hear you.
Okay. Okay. So my question was, being an attenuated virus, what is the impact of mutations? What is the likely impact of mutations, given that it's a particular, I'm not so sure about the science of an attenuated virus, but what do you suppose would be the impact in the event that we've got mutations in the community?
We can't hear anyone.
Yeah, thanks, Mark John, for that question. The live attenuated, this is a whole live virus that is weakened so that it does not cause disease. And this is the virus that the Chinese have been working on, and they've put on clinical trials in China. And I think it's now getting global acceptance. Now, the mutations that have been quite significant have been for the new technologies, right? Because they are based on the spike protein. So that will affect the efficacy of the spike protein. If you look at the lab studies with the new variants, right, the neutralizing antibodies with the Sinopharm vaccine have remained kind of, there's been very minimal impact on it. That's why the smile on that slide. Now, another benefit that you have with the live attenuated vaccine is then the body responds to other epitopes other than the epitopes on the spike protein, right? You now have immune responses or T-cell mediated responses to the M protein, to the N protein, and to other sites on the virus that were not targeted by these other vaccines. But still, you will need to monitor the impact of a live attenuated vaccine in immunocompromised patients, right? You know that they might be vaccine-induced disease. I don't know whether I've answered you there.
Thank you very much, Becky. Please, if there are any questions, go ahead. One or two more questions, go ahead and ask, so we can move on.
Hello, Dr. Mwale. My apologies. I can't see where to raise my hand there, but this is Nyarazo, Dr. Mgodi. Thank you very much, Dr. Magazi, for your presentation. You talked about neutralization. I just wanted to find out, do we, at the moment, do we have any neutralization marker that we can, an assay that we can use as a marker to see how a population is going to respond, or how effective a certain vaccine is going to be in a certain population? So I just wanted to find out if we do have any potential neutralization assays that we can use so that we are well informed. I'm talking from my background of HIV prevention. We are working on certain markers similar to that. So I just wanted to find out, in COVID-19, do we have specific assays or any potential assays? Thank you.
Thanks, Nyarazo. To the best of my knowledge, not at this stage. And like I mentioned earlier on in the presentation, in that basic immunology slide, is we are still at sea here when it comes to what are the markers of protection, right? What are the markers that we need to be looking at for protection against infection, against transmission, against disease severity, and against death.
Okay, thank you.
Hello.
Okay, thank you. Are there any more questions, guys?
Yes, Dr. Mari wants to talk. Hello, Dr. Mane.
Hello?
It's Dr. Mari from Australia.
Oh, okay. Please go ahead, Dr. Mari. Thank you very much. Go ahead and ask, please.
Oh, thank you so much. Thank you to the organizers of the webinar, and thank you to Dr. Magazi for the nice presentation. I've just joined it midway. My question is on the efficacy of the new vaccines. I can see your slide, which is on at the moment, which is puts Johnson & Johnson, the single dose, quite reasonably protected from the new variants. When you look at a country trying to buy one vaccine for the nation, what would you suggest is the best way to look at the different vaccines coming in, besides the storage, transportation challenges? And second question is, with already populations infected, how efficacious are these vaccines going to be vis-a-vis people who have already been infected and they've got some antibodies?
Thanks, Dr. Mari, for that. If we are looking at choice for a vaccine, there were those five considerations that I had put on in an earlier slide. To let me find right. These would be the 5 considerations that we would actually want to look at. Right, and safety paramount, because this is you are giving this to quite healthy people. And I must add here that what has been noted with the mRNA vaccines is you get more reactions to the vaccine than you would with other normal vaccines, and that's mainly driven by that lipid that you use as a carrier for the mRNA. So, but these are not severe enough to stop vaccinating with the vaccine. You will need to be very cautious when you, I see HTO has got his hand up, and he will tell us from the UK experience. You've got anaphylaxis that has been severe in a couple of cases. That's why the recommendation is to stay for about 30 minutes after getting it. And efficacy, and then there's the issue of cost, transport, dose regimen, and you develop a surveillance program. How should we introduce them? It's a question that has seized the South Africans this side, and what they have come up with is a phased-in introduction. You can't say this is the best vaccine at this stage, right? You can't say this is a global vaccine that will cover all populations. Remember, in infectious diseases, it's a function of 3 things. It's the organism, the host, and the intervention. So this organism is a moving target. It keeps on mutating. It keeps on resisting what you're throwing at it. So you will need to look at all those factors when you are choosing your vaccine, and there is no one better than the other. But you will need a surveillance program there. And then the other consideration, which is very important for our health systems, right, is, does this vaccine protect against severe disease? Does it protect against hospitalization? Because once it does not protect against hospitalization, I don't think it will be a very useful vaccine, because you then congest the hospitals with COVID, and then the other programs suffer. People with diabetes have no access to treatment. People with cardiovascular conditions have no access to treatment. Day surgeries here have been closed for the past, they only opened recently, last week or the other week. But since December, no day surgery or elective surgery was being performed. So those are quite key considerations when you are going to choose your vaccine. And at this stage, I won't say there is any better than the other vaccine. GTO will tell us about the UK experience and what they have done with these vaccines, because they've got an active program that's running there now.
I'll come to that in my lecture, Becky. Thank you very much. That was a fantastic lecture. The question I had for you is based on the governmental extensions of duration between first and second vaccination. What is your view, considering the studies that have been done and then governmental decisions to lengthen durations?
I was hoping you wouldn't ask that question. Right. I think in one of the clinical trials with, I think it's with the I think it's the AstraZeneca vaccine, where you got a more profound immune response if you delayed the second vaccination by up to 12 weeks. Right. So you actually have a benefit from a physiological benefit by delaying that. Then there is also a logistical a benefit in that you vaccinate a larger population. And what are the protections from the first dose? Now, the protections from the first dose vary from what vaccine you are using, right? Novavax vaccine and the and I can remember this vaccine, you get about 40 to 60% protection with the initial dose. So in a shipwreck situation where you've got a canoe with one oar, you cannot say, I can't use this canoe, I will rather have two oars before I use a canoe. So it's a delicate balance. There is scientific evidence to support the prolongation of the second dose. And there is a very big logistical reason. The factories cannot cope with the manufacture at this stage in the volumes that are wanted. And particularly now that you've got these variants emerging, right, you will start needing to alter the formulations of the vaccines. There is no perfect answer to that question, unfortunately, HB.
Okay. Thank you, Dr. Magazi. Thank you very much for your presentation. I think for the sake of time, we'll move on. I hope we have some time later on for more questions. And also, Professor Kerina Duri is going to speak. She's also coming from a scientific background. I'm sure she'll be able to answer some of the questions as well. So thank you very much, Becky. I think we'll allow Professor Duri to share a presentation, and then we carry on. Professor Duri is with the University of Zimbabwe. Her basic training was at UZ, but she went on to specialize in immunology and virology in Norway. So, Professor Duri, if you're there, please can you share your presentation and start?
Good morning.
Morning. Hello, how are you?
Thank you. Thank you for the invitation. Okay, thank you for the invitation. Mine is more of I was tasked to talk about how these vaccines work, more from a basic science perspective. I don't have much real clinical hands on COVID-19. My background, I'm working more on HIV. So mine is sort of really basic science talk. Thank you.
Thank you very much.
So it's sort of a refresher course. Yes, so this is my slide. Okay, so I wish to okay, so I just want to sort of to simplify this, the vaccine jargon, and also to understand how the 6 classes of more than 60 COVID vaccines work according to Dr. Mwale's excellent presentation. So I'll concentrate more on the 6 which are already on the market, how they work. So that probably, since in Zimbabwe, I think vaccination is voluntary, I don't know which ones will be brought in by the government, but I know of the Sino vaccine which will be coming, but for people to be informed of this different antigen preparation of these 6 classes of vaccines. So what I'm going to do, I want to talk a little bit about the background, which has already been covered by Dr. Mwale. I'll probably rush on those slides. And I think history is also quite important, because I want to also walk you through the early, what has been happening since Dr. Jenner's preparation and also how it is been revolutionized to the genetic vaccines now. I hope you are seeing my slides.
Yeah, if you could put on slideshow, please, Prof.
Okay, sorry about that. Okay, so and a little bit about the adjuvants. I think also Dr. Mwale. So it's also sort of overlapping, but Dr. Mwale was more clinical, and also some of the sticking issues, which I think is they are critical about also correlates of protection, stability, and also most of the things also issues have been also been discussed by the previous speaker. So probably I thought, okay, this is really basic about active immunization and artificial, which you want to talk more. Oh, sorry. I don't know what is happening to my slide. Sorry. Okay, the artificial one and the natural one, the natural immunization, which we get through natural infection, which I think is also a subject of interest, more so with this community transmission which we have. And this may also have a bearing on how our vaccines are going to perform in the artificial one, and also about the passive immunization of COVID virus a little bit, but not much on that one. Sorry, my technology is failing me a bit here. Okay. So like what I'm saying, more is the aim of vaccine development is sort of to alter the pathogen, like the inactivated or the weakened one, or toxins if we are talking about the vaccines from bacteria, so that it becomes harmless, so that it's able to mount an immune response to B cells, to T cells, so that we are protected, and in the event we eradicate diseases. So what is also important is also about specificity and memory. So if you can also see, I don't know how to, you can see my cursor, that also during the initial immune response, the primary immune response, we have this lag phase, which is very prolonged, which is sort of reduced during a subsequent a response to vaccines. When you get a subsequent encounter with antigen during actual infection, or then we have the lag phase also. So like also what Dr. Male said, we also talk about the innate immunity, which is genetic. We cannot do much about it, but it's also very important that we understand the innate immunity aspect, which not many people are looking at. Most of the vaccines, we are now talking more about this in the innate immunity aspect, which is in pink on my slides, about the natural killer cells and the complement, how it also affects this COVID. Not much work is being done. It's a lot of work now on the adaptive immune system, the B cells and the T cells, which I think is really commendable. It's a lot of work going on around that. But I think we should also look at the innate immunity aspect. So I've also talked about the coronavirus phylogenetics, but what I want to emphasize, which was not discussed, is the unique aspect of this coronavirus too, that the particles are shared during the pre-asymptomatic phase of infection as compared to the other human coronavirus, which makes it a bit difficult to control. And that's why we have this problem we have. Okay, the other thing is not, we've already talked, doctor had a slide on Zimbabwe cases. So, but what is important now, the vaccines are important because of this huge economic damage, and people are getting poorer more so in Zimbabwe, when people are self-employed, they have to move about, and also the social cost, and also the mental health issue aspect, which is also kicking in, is also important. So this issue of vaccinology to development is important, and the impressive speed which we've observed is very good. It has never been seen before, the speed of development of this vaccine. So I will not also dwell much about the spike protein. We've also, but I want to also emphasize the spike protein and how it is used to enter the cell, and what we have, the angiotensin converting enzyme on the host cell. We are going to, and also what is important here, since some of the vaccines use the subunit 1, the receptor binding domain, which also interact with this, is also important to facilitate viral attachment. So from the historical perspective, we also talk about Edward Jenner, about the smallpox. This is the first person who also developed in the 18th century, and how he used different vaccine, which we are already seeing even in the heterologous platform. That is 2 vaccine so we are using 2 vaccines to also come out with an effective vaccine. So in this case, he also used sort of a milder vaccine to raise an immune response to protect against the more virulent vaccine. So it's not something very new. Some of the preparations of using 2 vaccines, 2 viruses to come out with an effective vaccines. So that's the other thing. So using Dr. Jenner's finding, then we also talked about Pasteur, the French microbiologist, who also observed that this is the issue about attenuation, which you are also talking about, the sign of vaccine, which you are talking about. But of course, it's the processes of attenuation probably which are different. So in this case, during the 18th century, they just observed that time. They just observed that the old cultures sort of lost their virulence, probably due to mutations, such that when they try to make this chicken cholera vaccine, the chickens no longer died from the virulent strain. So the issue about attenuation, the issue about weakening the vaccine is not nothing, it's not anything new. So it has been done before. Probably it's the technology that has improved over the years. So by then the attenuation, so the viruses, like what I said, they are parasites, like the previous speaker. So they replicate so quickly, and the mutation rate is very high because of the polymerase enzyme, which it used with very reduced proofreading activities. So because we are also making this is what is also causing some of this mutation. So it's the mutation, and also passing the virus through different species is also been part of a strategy to weaken or to decrease the virulence of the viruses, sorry. So, and also can also use the recombinant DNA strategies, in which you can also isolate and also delete the virulence gene or mutate it so that it's now harmless. So you can also do more things in the lab, which were done, which took time, but when you do it in the lab, it also is faster. And like when you can also alter all these temperatures and also anything. And the good example also about changing the environment is the BCG vaccine, in which the addition of bile in the media also affected the virulence. So there's also a lot of science also to attenuate or weaken these viruses, which I think can be done. So I think these are sort of the different antigen preparation for almost all the virus which we have, including the COVID-19, the natural. Okay, you can have some immunity. Some of the viruses, once infected, like the measles, you are infected, you are protected for life. So for the natural infection with the COVID, that's, I think, the other sticking point. We don't really know. Of course, we know you are protected for at least about 5 to 6 months. That's what the literature is saying. So, but so we don't really know how of course the half-lives has not been sort of being researched around in different environment, because like what the host, the environment also plays a role in the comorbidities and also all these other things. So we have also a lot of issues around natural infection, more so around asymptomatic COVID infection. I think we should also look into that. So the attenuated, I think this is, we have some vaccine, which we have an example of COVID, and also the dead live, the dead, of course, I don't think we have a candidate there for SARS. Then, but the subunit down there in the recombinant, most of our preparations are recombinant DNA-based antigen production for the SARS, for the COVID-19 vaccines. So once you have this, your antigen preparation done, so there's also this issue about scaling up this antigen production in the purification, which previously we used to have production in animal tissues, which we now know it has been really a challenge to do with expense, keeping the animals, and the issues to do with the zoonosis. And I think a big example is this the polio vaccine, which was grown in monkey kidneys and was contaminated by the SV40, this monkey virus. And I think that's why we've moved from this animal tissue to human cell lines. Because if you also look on misinformation on media, you also see a lot of people also talking why we are also using these human cell lines. So most of also our vaccines, including the Sino, the Oxford, the Seneca, they also use these cell lines from the HEC and also the MRC and so on, which are also from aborted fetuses some years back. But this also raises a lot of some ethical issues with some, probably the Catholics and some anti-abortive movements. But what is important about these cell lines, they sort of there'll be small factories to sort of to produce these quantities of virus antigens in big quantities. So, and also then you have also to purify them so that we don't have any contaminants of human tissue in the vaccines. So it's a lot of, and it's quite expensive. But the more we purify it, we also run to the issue of losing immunogenicity of these antigens. So that's when the issue of adjuvant, which was also mentioned by Dr. Magazi, kick in. So the adjuvant, so we want to increase antibody production. So there are different type of adjuvants, depending, some just oil in water, or you can put some mycobacterium some mycobacterium constituents there, and aluminium. I think that's the main one, the aluminium one, the alum, and also some can also have some immune stimulatory complexes, sort of to boost the immune response. So the types of COVID vaccines, basically I classify them into 6. The messenger RNA vaccines, non-amplifying. Then you can also have the self-amplifying messenger RNA vaccines. But now there's none of this, the self-amplifying one is now on the market, but I think you'll see why it's also important. Then you can also, the viral vectors, Dr. Magwazi has talked much about those ones, and the protein subunit vectors. Most of our vaccines are protein subunit vaccines. I think a majority of our vaccines and potential candidates are around the protein subunit vaccines, and the inactivated whole virus vaccine, the Sino, and the DNA vaccines. We don't have anything much on these ones, but also for the RNA and the DNA, it's also important to remind you where transcription takes off or translation. When the DNA transcribes to DNA, or what happens, the protein the protein synthesis is done in the cytoplasm, so nothing in the nucleus, whereas there are so many myths about whether the messenger RNA can also be incorporated into the host genome. So then when we talk about the mRNA vaccines, Doctor has already talked about about it, so I will not take much of your time, but to highlight the aspect of lipid nanoparticles, which are also embedded on the messenger RNA to make it a little bit stable. And also, the lipids also act as adjuvants. So then, like what I'm saying, is the translation aspect. We don't need to care about the nucleus and this aspect about this integration of viral genome into the host genome. And this is quite faster and cheaper. It can be done into the lab, and it can also be easily modified, the mutate, to accommodate these mutant strains, as previously highlighted by the previous speaker. So I think this is what I'm trying to say. So if we have your gene sequence, whatever variants, the UK variants and other variants to come, we just package them in these nanoparticles and then express them without taking much time. So I think it would be much easier to cater for the mutants, strains of the COVID-19 virus. So these are the examples, like what he said, the previous speaker, the Moderna virus and the Pfizer are based on the messenger RNA virus, but of course there's some mild adverse reactions, and also which may occur following the booster doses. So I thought it was also nice to give the examples for, then there are also others, there are so many in candidates, messenger RNA-based COVID-19 candidates still being researched on all over the world. But you can also see, I don't know whether it is this other, some companies that has also produced a MERS virus based on the messenger RNA, also still working on the COVID, the Japanese one on this slide. So then we also go to the other classes, the viral vector vaccines. It's also, most of them, they are based on the viral vector, mainly the adenovirus. So it's also not new. The viral vectors started as early as the early 1970s. And we already have some vaccines already using this, preparation, the Zika, the flu, the Ebola virus. So nothing is really new other than the messenger RNA. So you have the spike protein. So it has been made harmless by deletion of this early region, which makes it able to replicate. So it's quite safe. And you can also have the other more effective adenovirus, different strains, to make it more harmless and safer to deliver the immunogenic antigen. So can also have examples of vaccines using the viral vector. It's already saying, we've already talked about it. And the issue to overcome the pre-existing immunity against this adenovirus following upper respiratory infection have already been discussed. And that's why you also have this heterologous boost strategy in which you can also use a different adenovirus from our cousins, the gorillas, the chimpanzees. Then you can also see that the AstraZeneca virus vaccine used the chimpanzee boost also to circumvent this challenge of pre-existing immunity. So the advantages also, these viral vectored vaccines, they can be, the expression of protein is quite high and the induction of the immune response is quite strong. And also, like what I said, is not, they do not affect or interact with the host DNA, which is a myth, also of misinformation which is circulating. So the inactivated vaccine, so it's also the Sino. So they've inactivated the virus, but it's also nice because, of course, if it's inactivated, we are not concentrated on the spike protein also. We can also get an immune response to other antigens that which is not really been so far been researched. So I think it also leaves room for us to can get other immune response. So it's nice as we get this sinovirus also to do some research to see the titers of antibodies and the specific antibodies produced after administering this whole inactivated virus. But as the previous speaker said, it's not appropriate for people who are immunocompromised. So these are also more examples of inactivated COVID candidates. It's a lot of them in preclinical stages. But okay, but also for the KM biologics, they also have the inactivated virus. We have the Zika virus, which is also already on the market. Then the attenuated virus is also some more examples. Then the protein subunit, okay, so it's also another example, which is the synthetic subunit approach lies in between the nucleic acid and the inactivated virus vaccines. So it also recombinant and also typically also expressed in cell lines, including the insect cell lines. So we have this Novavax vaccines as an example for that platform. So it's a lot of all this still in preclinical development, a lot of candidates. Then you can also have other classes which are not yet on the market, the DNA viruses, and also more interestingly, the virus-like particles in which we are using informatics or bioinformatics analysis to map sort of the immunogenic, the epitopes and the immunogenic response so that you can also produce precise the antigen that are more immunogenic. So it's also a new, so we have also a lot of candidates, a vaccine using this. Okay, so this is an example of the DNA platforms, already candidates using the DNA and the then the concern about the DNA vaccine, which says there are some concerns that it may be integrated in the host chromosome and may turn on and off some tumor oncogene or tumor suppressor genes. Then these are examples of the virus-like particles. It's a lot of also some virus candidates also using this platform. Then also, I thought it's also nice to talk a little bit about this, some sticking issues in this COVID vaccine development, the issues to make the current messenger RNA virus more stable, more so here in the developed country. I think it's a pie in the sky to have them if they need these -70 temperatures. So I think we also have a lot of research around additives that may also being used to extend the vaccine shelf life, and also to have it lyophilized or freeze dry them so that you can just reconstitute a powder before injection. So we have a California-based company also working on this strategy, and also to make it the shelf life easier. I mean, that's the logistics. Then the issue about we talked about the messenger RNA, but there's also the issue about the self-amplifying messenger RNA, in which the self-amplifying, like what he's saying, it's also used in this plasmid, the Venezuelan equipped encephalitis virus. So what is it? Why is it important? It's also important because we may also run away from all these second doses or third doses because it will be self-amplifying. And also there's also the issue to reduce the amount of vaccine or the volume of vaccine that can be administered into the arm. So far, like the messenger RNA doses range from 30 micrograms for the Pfizer vaccine to 100 micrograms to the Moderna virus. So it's a lot of, the mass is quite huge. Then you can have this CureVac, which is 30 microgram. So what we are saying about this self-amplifying messenger RNA can also overcome these issues to do with the high volume of vaccine doses. So the vector is engineered so that it can also make more copies and it self-replicates. So that's the other issues. Then the issue about circulating variants is also an issue, like what I said, the viruses are very unstable, mutating. So the previous speaker has already talked about this virus, so I will not take much of your time on this one, in this slide. But also in Zimbabwe, I also understand we also have some surveillance system which also being put in place at our national reference lab in collaboration with our UK partners, which I think is also very important, is these mutants also like the potential consequences is also being discussed. So I feel I'm also repeating ability, transmissibility, severity of diseases, and also these issues to do with false negatives, which I think is really an issue so far, even in Zimbabwe. I mean, most of even my friends and my families with clinical disease, but they have false positive PCR results. And also, we can also have this issue about evasion of natural or induced immunity because of this immunity, of this mutation. So this is also quite a concern, given that we are vaccinating a large proportion of the population globally. I think it will be also an issue in the long term. And also, how long, okay, the other issue is also, some people are also very skeptical about how fast the COVID vaccine has been produced compared to the traditional other vaccines, which is an impressive 1 year compared to the polio vaccine that took almost 50 years, and measles 30 years, and so on. And also some of the infection like HIV is 40 years now, and we are still not, we are still going, we don't have any vaccines. So I think I also get some misinformation about how also people think, why is it so fast we got this COVID vaccine in 1 year? But I think it's also due to that the COVID vaccine development, because it's a global problem, it was well funded and it was really produced and very fast. And also, there are also some issues about the use of human cell lines, the fetal one, more so to do with the MRSA, sort of the aborted fetus cell lines. So the Catholics and some people may not also be willing to take it up. So I thought it's also nice to say, I mean, depending on your belief and so on, these are some of the things which some people may also want to consider before they choose their vaccine of interest. Because since here also in Zimbabwe, it's also voluntary. And I hope we can be obvious, we will have the opportunity to choose if we have the option. So this is what these are the features of an effective vaccine. So what is now important is if we can also have a tick box. So about safety, safety and protectiveness. I think this, since they passed the phase 3 trial, the phase 3 clinical trials, the issue about safety and protectiveness, it's a tick and a tick there. But the issues to do with sustained protection. So I think this is the challenge which we have currently with our vaccines. Of course, we know they are effective 95%, whatever, but the longevity, how long we will be protected, we still don't know. And the correlates of protection, we don't know. And so the issues to do with the antibodies and the T cells, I think also the previous speaker touched on that. And also the practical aspect about the cost, the biological stability, ease of administration, the storage and transportation, and the side effects are some. Because I heard someone who was asking about which criteria which we can use to select the vaccine which may be suited for our situation. So I think this slide may also partly answer that question. So, and also this issue to do with herd immunity. I think it's also important that I think it's also not necessary to vaccinate 100%, because if we reach about 70%, we have reduced the transmission rates. The virus is less efficient to transmit, and eventually the infection will not maintain itself. And this is what is sort of what we mean when we talk about herd immunity. So I think which is a good news. So since we don't have to vaccinate everyone, so who is to be vaccinated and why and how, I think is also the public health aspect, I think, but it's obvious, targeting the people who are at risk and so on and so on. So I think this is the end of my presentation. Thank you.
Thank you very much, Professor Luri. Thanks for teaching us the science of these vaccines. I think it's very important that we understand the basics about the vaccines for us to make good decisions. So let's now move on to questions. If there are any questions for Prof, please go ahead. Yes, Mr. Macheka, you can go ahead.
Thank you very much, Prof, for that very interesting presentation, which takes some of us slow learners through the basics of this vaccination process. Your very last slide touched on herd immunity, and the previous one was talking about the Sinovac, and that is an inactivated virus, and that is deemed unsuitable for the immunocompromised. The problem with that is sometimes it's taken as a very loose term. When you say immunocompromised, is there a level of immunocompromise where you say this one, for example, with your experience with your HIV cases, who would you say can receive it and who would you say cannot receive it? Or is it that all those who may potentially be immunocompromised must not be exposed to that kind of virus and rely on herd immunity? Thank you.
Thank you for the question. Yeah, I think immunocompromised, I think we can classify for HIV, I think we'll be talking of those people with AIDS. I mean, with the CD4 count probably less than 300 or so. I'm not sure whether there have been some studies which you can also attest to that. But if you are really sick, and I think we should have sort of a cutoff CD4 when, but now I don't know for HIV, I think we have sort of a cutoff for the CD4 count if it's less than 350, I think 300 to 350, or some patients 250, I think, or some patients on cytotoxic drugs, or the cancer patients, probably they should also be discouraged probably to take those the vaccines. Yeah, I think probably that's what I can also say. But if you are immunocompromised, but you are still immune competent, yeah, because it's about if your immune system is still not that down, I think you can still take the virus. Just like even our babies, the infected babies, HIV infected babies, they still take these IPI vaccines, and I think they are still doing very well.
Okay, thank you very much.
I'm not sure whether I answered your question.
Yes, I think it did clarify some issues for me. Thank you very much.
Hello, so are there any other questions?
Ichikari, just to add on what Prof has just said, the issue is when we say with live attenuated vaccines, you should use them with caution in the immunocompromised. The thresholds for immunocompromise are not exactly well defined, but the concern there is one, you might not get the immune response or the neutralization antibodies that you desire because of the immunocompromised state. And two, you might get vaccine-induced disease. And with the current attenuation processes, the second concern does not falls away, but not entirely. So that's why we say use with caution. We've got this experience from yellow fever vaccination, particularly here in Africa, where we've got high HIV burdens. We still vaccinate people against yellow fever without much of and achieving good antibody titers and without activation of yellow fever disease. So it's a cautionary statement that you use with caution in the immunocompromised for those reasons.
Sure, thank you for that, Becky. Thank you for the clarification. Okay. Right, so thank you very much, Prof. I think we'll move on. For the sake of time, let's move on and with our next presenter. Egidio da Silva is a consultant anesthetist in the United Kingdom. He is the son of the soil. He trained here initially at the medical school, and then went on to the UK to train in anesthesia and perioperative medicine. He was involved recently in a program to administer vaccines to, I think, some 5,000 health workers in the UK. So he's got a lot of experience with clinical handling of the vaccine issues. So, Jido, if you